everyone. Thank you for joining us today. My name is Stacy Schultz-Cherry, and I am the co-editor-in-chief for the Journal of Virology, or JVI. The American Society for Microbiology publishes the Journal of Virology, which is hosting today's webinar, as well as all others in what we call the Journal of Virology seminar series. For more information on the series and to watch our introductory episode or our previous episodes, please visit the Journal of Virology website, or you can scan the QR codes you see on the slides. So if you're not, there you go, there are your QR codes. Okay, next slide, please. If you're not familiar with the Journal of Virology, we publish really preeminent work in virology research. We welcome everything, primary research, mini reviews, and other article formats that really interrogate fundamental processes and structures in viruses, the mechanisms by which they interact and evolve with their host and environment, and novel methodologies, therapeutics, and diagnostic strategies. Again, we are all inclusive, all viruses, really all processes. Next slide, please. Our webinar today is entitled Blocking the Competition, Superinfection Inhibition by Giant Viruses. Our speaker is the first author of the paper that was published in the Journal of Virology in September 2024. If you have not read her paper, I highly encourage you to do so. It's free and accessible from the Journal of Virology website. Now, before I turn things over to our speaker and our host, let me quickly go through the format of our webinar and some housekeeping rules. So the first 35 minutes is all science, right? Speaker introductions, scientific background, and really going through the paper. And then we're going to take some time and talk a little bit about science advocacy. Why are the studies important? What do they mean for your neighbors, grandparents, the public? And we'll also ask Isabella to share her experience in going into science and things she wants people to know about her career or the work that she's doing. And then we're really going to open it up to you guys to discuss the manuscript and question and answers. The webinar and future Journal of Virology seminar series webinars are recorded, posted on the Journal of Virology website, and we are hosting these every other month. So at the end, I will announce our September, at least the paper for our September webinar. In lieu of a certificate of attendance, you will receive an email for registering for this webinar. It serves as proof of your attendance. We do not provide certificate of attendance. If you have any questions for our speakers, throughout you use the question and answer button at the bottom of your Zoom screen. We're not using the chat function, so please submit all of your questions and comments via the QA button. And if we don't get to all of the questions, we'll share those with Isabella and she can decide what she wants to answer for the future. All right, now I'm going to invite our host with the most, Dr. Kristen Parent, to introduce our topic and speaker today. Kristen.
Thank you, Stacy, and welcome everybody. It is an honor to introduce Isabella Aquino. She's going to be talking to you about a really fascinating group of viruses called giant viruses. As you may guess from their name, they're very, very large, the largest on the planet. And as you'll hear about, they're really enigmatic, being one of the newest discovered viruses with many genes that we really don't know what they do. So this is also very challenging work because giant viruses have very little toolkits available in terms of genetics or biochemistry that we have to study other viral systems. So we have to be very creative to understand their life cycle. And you're about to hear some really beautiful work understanding how giant viruses take over a cell and keep out other competitors from infecting the same cell in a really beautiful study. So welcome everybody, and welcome Isabella. We're really looking forward to hearing your talk.
Thank you, Kristen. And I will start my presentation now. So can everybody see my screen?
Nope, not yet.
Okay. Okay.
Perfect. Thank you.
Welcome. So virosphere is extremely complex, and viruses are the most abundant entities that we can find in the planet. And we estimate that the number of the viruses that we can find in the world is bigger than the number of the stars that we can find in our galaxy. And among them, we can find the giant viruses of amoebas. They are known because they have like characteristics that goes in a opposite direction for what we used to see for canonic viruses. For example, the size of the particles, they show so much bigger particles than when we are compared to canonic viruses. And this allows them to be seen by optical microscopy and also to be retained in filters containing pores of 0.22 micrometers. And also they are able to carry more than one type of nucleic acid in their particles. And among those genes, we can find transcription and translation linked genes. And also when we are talking about the giant viruses host, it's a very different one from what we used to see. The amoebas, they are considered the fossils of eukaryotes. And those viruses show a big heterogeneity of morphologies and also about their genomes, showing a wide diversity of viral particle morphologies, as we can see in this image that I'm showing you guys here, where we can see oval-shaped particles or particles with more complex structures, like the case of the Superinfection virus, for example, round-shaped particles, and also capsids with the icosahedral symmetry. And we also can see different genome sizes, and they can reach until like 2 million of base pairs. And they also show a very diverse genomic content, so we can find a lot of different genes inside those viruses' genome. And this variety is not only something that we can find among all of the giant viruses, but also in some groups of giant viruses, like, for example, mimiviruses. I will refer to those 3 different viruses here, like mimiviruses, like we used to do before the last taxonomic classification of giant viruses. But I'm speaking here of mimiviruses like Acanthamoeba polyphaga mimivirus, or APMV, the first giant virus isolated in the world, and also moumouvirus and megaviruses. So they show a similar morphology of the particles, for example, but they can also show a lot of differences among them. So one thing that is very similar, but is also different among those viruses, is the presence of the surface fibrils covering all the capsid. And these fibrils can be different in like the number of fibrils or the length of the fibrils, and also like in the way that they organize themselves surrounding the capsid. And after the first giant viruses isolation, many giant viruses were isolated across various geographic regions and ecological niches, including here in Brazil, where in the last 14 years of Brazilian studies, especially by our study group, Giant Viruses Study Group, that is led by Professor Jonatas Abraão, we had the opportunity to isolate a lot of different giant viruses of amoebas. And for example, the first isolated here in Brazil was sambavirus, and sambavirus is a mimivirus, but we had a lot of different giant viruses being isolated before, like the case of Marseille viruses, or also very different viruses like Superinfection virus or Pandora virus, and a lot of others. And I can cite also one that is very different here, that is Yara virus. And this shows for us a huge variety of giant viruses that were analyzed and isolated here in Brazil. And many of them come from a common source. We already had the opportunity to analyze so many samples from all over the country, but a lot of giant viruses that were isolated came from samples collected in Pampulha Lagoon. Pampulha Lagoon is considered like a postal card from my city, Belo Horizonte. And a lot of samples that we collected here were already analyzed, and a lot of different viruses were isolated from Pampulha Lagoon. But I think something that really caught attention of a lot of people is how this isolation happens. What are the methodology that we use to do the isolation? So I will briefly speak a little bit about how we do this. And first we have to collect the samples, and the samples can be water samples or sewage or mud or even soil samples. And then we are going to pass through the to the process of processing those samples. And for the ones that are already liquid, we are going to do some liquids and dilute those liquids. And in the case of those that we are going to find something solid, like in the case of mud or soil, we have to put a little bit of water before and wait about 24 hours for the decantation of those samples. And then we are going to pass through the process of doing the dilutions and the liquids. And then we're going to storage those samples at minus 20 Celsius degrees until the time of until the time that we are going to use the samples to do the process of prospection. And for the prospection, we use microplates of 96 well plates, 96 wells, I'm sorry. And inside of those wells, we put amoebas. We do a culture of amoebas there. And then we come with the liquids and we put the samples into those amoebas monolayers. And every sample is going to be analyzed in duplicates. And then we are going to wait for about 7 days the most and analyze daily all those wells of our plates to look for the presence of some indicative that maybe we have a presence of a giant virus in the culture. And what is the indicative that we usually look for? We look for cytopathic effects, and the most usual to see in the case of giant viruses is the rounding of the cells, and we also can see the cells losing from the monolayer, and also lysis, and a lot of those can indicate the presence of a giant virus. And to confirm if this is actually a cytopathic effect or not a cytotoxic one, we collect those samples where we saw the cytopathic effect, and we pass through a flask. And in this flask, we're going to put again the monolayer of amoebas and then inoculate our sample. And we do this because it's a bigger space, so the sample is going to be diluted, and if is something causing a cytotoxic effect, this is not going to happen inside the flask. So after the confirmation, we start to really analyze those samples to see if there is really a giant virus there. For example, we analyze them for transmission electron microscopy and other types of microscopy and other types of analysis too. And during our efforts to do new isolations here in Brazil, we isolated the new megavirus, and it was called megavirus caipurensis. And this virus was also isolated from water samples collected in Pampulha Lagoon in the year of 2016. And something that caught our attention to study a little bit more about those viruses was the unusual surface fibrils that they present in the capsid. They organize themselves, forming those little groups of fibrils, those little clumps, and this caught our attention. And that's why we started to do genomic morphological characterization of this virus during my master's. And during this time, when we studied a little bit more about the genome of these viruses, we confirmed that it was a megavirus. And we saw a very similar characteristics to other megaviruses, like the size of the genome and also the number of ORFs that we, or the genes that we are finding inside the genome, and also the size of particle and things like this. But how all of this work here started? So when I was analyzing all the replication cycle of this virus, we saw something that was really weird for us and that caught our attention. That was observation of vesicles that were enclosing newly formed particles of megavirus typorensis near of the viral fabrics. And those viral fabrics were producing megaviruses, new particles. And why this caught our attention? Because it's not usual to see this kind of vesicles in such late stages of the replication cycle. This is more usual to see like phagosomes in the beginning of the cycle after the virus is phagocytized by the amoeba. And we started to think that maybe those vesicles could be linked to the capacity of those viruses to exit the cell using the process of exocytosis. And this was never reported before for mega, moumou, or mimiviruses, so we decided to investigate it. And how we did this? So the first experiment that we did was infecting amoebas with mimi, moumou, and megavirus. And also we counted the number of viable cells in the system, amoeba more virus. And what we saw was that the viral titer and the number of viable cells was inversely proportional for the three viruses. And what this indicates for us? This indicates for us that if you have a decrease of the number of viable cells in this system, but you have an increase of the number of newly formed particles, these cells are probably being lysed, they are dying, so they can, so it can happen the exit of this virus from the host cell. So this is not what happens in the case of exocytosis, where the cell doesn't actually needs to be lysed to so that these particles can exit the cell. And when we analyzed a control experiment that was made with Cedra virus, there is another giant virus known for having the capacity of exiting the cell, also using the exocytosis. We can see that the increase in the viral titer is not linked to the decrease in the number of the viable cells in the time of 4 to 12 hours post-infection. And this shows us that those cells are not dying to liberate and to make the exit of these viruses from the cell. And moreover, when we analyze it using transmission electron microscopy images of amoebas that were infected with Cedra viruses, we can see a lot of particles within the exosomes, the vesicles that are used to do the process of exocytosis 8 hours post-infection, showing us that the exocytosis was not happening for megavirus. And then we decided to study the hypothesis of the superinfection. And what is the superinfection? So superinfection is the process where happens the entry of more than one virus or of the same or different species inside the whole cell. And some viruses are capable to inhibit the superinfection by blocking the entry of other particles at early stages. And why it is so important for some viruses to inhibit superinfection? Because it genetically favors the particle that enters first and also avoids intra and interspecific competition, and also leads to proper generation of viral progeny once that the cell is focused on producing new particles of only one type of virus. And this was already saw before by HIV viruses or for bacteriophages and also for another nucleocytovirus. So giant viruses of amoebas are nucleocytovirus, and they are closely related to vaccinia virus. But vaccinia virus are capable to infect animal cells. And what we can see is that vaccinia virus are capable of blocking the superinfection during the early expression of two different proteins, the A33 and the A36. And they are not only capable of blocking the superinfection, but also to expel these enemy viruses by the actin tails for the interior of the host cell, avoiding the competition. So when we observed this for another nucleocytovirus, we started to think like maybe it would be possible to find the occurrence of the superinfection phenomenon and or its inhibition among giant viruses of amoebas. So for this, we decided to study a little bit more about this hypothesis, designing an experiment where amoebas, Acantha amoeba was infected before with moumouvirus, and also megavirus. And the cells in this image can see in red. And 2 hours post-infection, we did an input with a different giant virus. Orpheovirus purified particles were used as baits in this experiment. So why we did use orpheoviruses in this experiment? Because they can infect Acantha amoeba. They are not capable of infecting productively these amoebas, and the particles are also really big. They can reach until 1 micrometer of length. So this was going to make more easier for us to see those particles when we were analyzing the immunofluorescence microscopy images. So after doing those 2 infections, we analyzed those images, and we could see that a lot of particles were phagocytosed by the amoebas that were previously infected with megavirus. And this is different from what we observed for mimi and moumouvirus. And also inside the vacuoles of the amoebas infected previously with megavirus, we can see not only 1 particle, but more than 1 particle inside those vacuoles. And when we try to quantify this number of cells with phagocytized baits in the interior of the vacuoles, we could see a significantly higher mean of phagocytosed baits for the cells that were previously infected with megavirus. And we could see also a bigger variety of the number of baits that were phagocytized by the cells that were infected first with megavirus. For example, for one cell, we could find 6 particles of orpheovirus inside the vacuoles. And we also did a titration of the residual orpheoviruses in those systems using an amoeba that these viruses are capable to infect productively, Vermamoeba vermiformis. And we could see 6 hours post-infection that a large part of the inoculum in megavirus was not recovered. And what this means? So this means that if we don't find a lot of particles in the supernatants of these infections, it means that those particles were phagocytosed by the amoeba, different for what we see for mimi and moumouviruses, showing us that the megavirus Caiporensis isn't capable of inhibiting the superinfection, but mimi and moumouviruses are capable. And moreover, when we observed by transmission electron microscopy images of the system amoeba, more megavirus, more orpheovirus, we can see particles of orpheovirus in the cytoplasm of the host amoeba near of viral fabrics that are producing new particles of megavirus. And we also did a titration in different times of infection of this system containing amoeba, virus, mimi, moumou, or megavirus, and orpheoviruses. And we could see that 2 hours post-infection, mimi and moumouvirus inhibit the superinfection because we have a reduction of the bait incorporation. And in this graph, we can see Mimi and Momo in the lights with square and a circle. And when we analyze the case of megaviruses here marked for a triangle, we cannot see this. And actually for megavirus, 12 hours post-infection, the baits are still being incorporated, showing again this incapacity of megavirus to inhibit the superinfection. And which mechanism is used to block the superinfection in giant viruses of amoebas? So when we analyzed IFM images of amoebas that were infected with the 3 viruses, analyzed it in the work, and compare them with the uninfected control, we can see that Mimi and moumouvirus presents, the cells presents a size reduction, conformational changes, and also the vacuole reduction. But when we analyze the megavirus infected cells, we can see the maintenance of the size when compared with the uninfected control, and also the maintenance of the amoeboid contour, and also the number of vacuoles. And when we try to quantify this, we saw that it happened the maintenance of the vacuole number for the cells that were infected with megavirus. And also when we measured the cells that were infected with the 3 viruses, we saw the maintenance of the cells size also for those that were infected with megavirus. And also we analyzed the emission, the capacity of the emission of pseudopods of those amoebas, and we can see that it happens a reduction of pseudopods in amoebas that were infected with mimivirus or in moumouvirus, but it doesn't happen in the case of amoebas infected with megavirus. And this demonstrates to us the inhibition of the superinfection happening because of the process of blocking the phagocytosis. Because to have the capacity of have a pseudopods, and also the maintenance of the number of vacuoles, and also the amoeboid contour and the size of the cell, this is all very important for the process of phagocytosis happening for the amoebas. And we see that all of this is passing through an inhibition in the case of Mimi and moumouvirus. So when you block the phagocytosis, you also can block the superinfection. And what are the consequences of megavirus superinfection in amoebas? So when we analyze the transmission electron microscopy images of more late stages of this replication cycle of megavirus, we could find a lot of late stage uncoating of phagocytosis particles in several empty capsids in the cytoplasm of the infected amoeba. And this was also something really weird because the process of uncoating happens in the beginning of the cycle and not in the end of the cycle. So the amoeba is going to phagocytize the viral particle, and then the viral particle inside the cytoplasm is going to suffer the uncoating, and the cycle is going to start from there. And this was something really weird for us. So we started to think about this to try to understand what was happening. And we came to the conclusion that this process was leading to a loss of part of the progeny that was formed, leading also to a lower number of particles that were available in the supernatant of cultures that were infected with megavirus. So maybe this is something that is a way of those amoebas try to protect themselves and the neighbor cells. Because if you have a bigger number of progeny formed that is available in the environment, you can reduce the chances of infection of neighbor cells. But we have to go deeper in this hypothesis to understand this a little bit more. And we also analyzed it if the superinfection could be chemically reversed. And for this experiment, after doing the infection of cells with mimi and momo and megaviruses, we treated those cells, those cultures, with 3 different chemical inhibitors. And one of them I will highlight here, there is cytokalazin. So cytokalazin is known as an inhibitor of phagocytosis. And in the case of amoebas that were infected before with mimi and moumouvirus, we didn't observe no changes after the treatment. But for those amoebas that were infected with megavirus, we can see changes. We can see increased titers after the treatment with cytokalazin in the case of these viruses. So if you are capable to chemically reverse the phagocytosis, you can also chemically reverse the superinfection, showing again how central the phagocytosis is for the process of inhibiting the superinfection in giant viruses of amoebas. So summarizing, we demonstrated in this work for the first time the existence of the superinfection process by giant viruses that infect amoebas, as well as its inhibition. And mimi and moumouviruses induce superinfection blockage in amoebas by inhibiting phagocytosis, leading to reduced cell size and suppression of vacuole and pseudopodia formation, like we can see in those images that I've showed before. Megaviruses are unable to inhibit phagocytosis and consequently the superinfection, allowing the continuous incorporation of newly formed virions, which negatively affects the available viral progeny. And despite their evolutionarily relatedness, mimi, momo, and megaviruses exhibit profound differences in their interactions with the host amoeba. And these findings reveal not only the functional diverse among related viruses, but also the sophisticated control mechanisms that may shape the viral composition and microbial ecology, and possibly aspects of eukaryotic evolution. And moreover, amoebas and their viruses are considered models for the first eukaryotes. So understanding this relationship can help us also to understand the origin of complex life on Earth. And what about the next steps? What we thought about doing to complement the results that we had in this work. So we think about explore the superinfection in other amoeba viruses, including those that enter using mechanisms other than phagocytosis, to see if this happens not only in other giant viruses of amoebas, but if different mechanisms can be involved with the inhibition of superinfection giant viruses. And we wish we can analyze which genes are involved in the inhibition of superinfection in mimiviruses. I showed earlier that two genes are central for the inhibition of phagocytosis for another nucleocitovirus. So we wish that we can dig in a little deeper and analyze what can be the genes that are involved in superinfection in mimiviruses, and to delve into mechanisms that govern the inhibition of phagocytosis and consequently the inhibition of the superinfection. Maybe some kind of signaling molecules or something like this that can be involved in this mechanism, and try to understand a little bit more this mechanism. And also assess the nature of superinfection in megaviruses, including the possibility that it represents an antiviral mechanism of acanthamoeba. We have in the literature a lot of studies, I'm exaggerating a little bit, but some studies that shows antiviral mechanisms for amoebas. And we can see this for acanthamoeba and also for vermiformis, and maybe try to study this a little bit more, looking for this size of the prism is going to be something interesting for us also. So that's it, and I would like to thank the American Society for Microbiology and the Journal of Virology for the opportunity to be here today presenting this work. and it's an honor for me to be representing all the collaborators of this work, and also to have this opportunity because I admire so much journal virology. It's a pleasure to know that I have some papers published in a journal that I admire so much, so thank you. And I would also like to thank all the collaborators of this work, UFMG, the Virus Laboratory, and also Professor Jonathan's Giant Viruses Study Group. I spent 7 years of my life working with them, and a lot of experiences that I had were like amazing, and everything started there. So thank you so much. And I would also like to thank the Microscopy Center of UFMG. All these images that I showed you guys from this paper and from a lot of different papers that were published by Giant Viruses Study Group were obtained in the Microscopy Center of UFMG. So thank you for the collaboration, all the help in our analysis. And I would like also to thank the financial support of the funding agencies here in Brazil that makes possible for us to do science. And of course, I want to thank you so much for everybody that is watching. Thank you for the audience, for the interest in something that I love so much, that are the giant viruses of amoebas. And I will leave here my contact and also the Professor Jonathan's contact. Feel free to get in touch with us for any kind of questions or something that I can answer here today, or curiosity. So thank you so much.
Thank you, Isabella, for the beautiful presentation and the work. It's just so exciting to see what you guys are doing. And I personally love the name of the giant viruses. They make me smile every time I hear them. So I think this session is really thinking about the general public, parents, grandparents, why does virology matter? So Isabella, this is just, we'll take about 10 minutes or so and just think about, tell us why the studies are important and, how you would explain them to somebody of what it means. And then a little bit about you and why you went into science and what you think are the most important or unanswered, the coolest unanswered questions in your field. So why are the studies important?
So Stacy, I thought a lot about this all of those years that I am involved with science in my life. And I think in the case of this study, we have to think about the importance of basic studies in virology. I've been working with basic virology since the beginning of my career, and just now I started to work with more applied virology. And I think we have to remember that if you don't have like the basic information, you can never do the applied science. So basic studies are important because they reveal like fundamental mechanisms of viral biology, for example, and all of those informations can have important implications in microbial ecology, for example, or evolutionary biology. So I think this work fits perfectly in what I'm saying right now. I think we have to remember this importance of basic studies. And when I think about the importance of science for my family, my friends, people that are linked to a more close circle of my life, I think the major thing that is important, especially for my parents and my family, is because here in Brazil it's not so easy to get the education and to get a PhD. So this is a huge opportunity for people just like me. I came from a family with humble roots. It was hard. It was too hard for me to have the opportunity to study, and this was the dream of my parents. So I think to see their daughter having the opportunity to become a scientist is something really important for my family and friends, even that sometimes they doesn't understand perfectly what I do. They feel really proud of me, and I always try to explain this for them, like this maybe is something weird for them, the giant viruses, and sometimes people get scared like, oh my god, giant virus, this can be something horrible for us. And I say, guys, when we think about microbiology, we have to think also not only in the side that is linked to the human health and animal health, but also a lot of those entities are more important for us to understand the evolution of life, the complex evolution of life in the earth, and also for like giant viruses has also a big capacity to be applied for biotechnology and different things. But I think the most important thing about them is to understand the evolution of viruses and also to understand ecology and things linked with something like this. And I think I also came into the world of science because since I was a child, I was always really curious. And all the time that I asked something for my parents or for someone, some adult, and I didn't get the answer, this caused me so much like frustration. And I think like, why I am a child and I am thinking about this, but anybody thought about this before? So this was something that I always wanted to do. I was always trying to get the answers that I didn't have. And when I came into the university, like I said, it was hard for me. So I wish I could leave everything there. So when I came to the first laboratory where I had the opportunity to work a little bit with science, I started to be like, to really feel amazed by this world. And I met Professor Jonathan when I was doing a class of him in I was studying microbiology in the biology graduation. And when I saw the first time a mimivirus, that just rocked my world because they are so different. And I said, oh my God, I never saw something like this before. I have to work with this man. So I had to tell him that I wanted to do this. And it was something that really was a game changer in my life to have this opportunity to get into science and to work with giant viruses also. So I think the thing that was more important for me for this was all the curiosity that I had in my life and all the answers that I was looking for. And I think when I think about the unanswered or most important questions in our field, when we think about giant viruses, we are always thinking about limits because I don't know. I think about the limits of virusphere. Is there a limit? Because every day we have a different report of some bigger virus or something really unexpected about the genome, and I don't know, something unexpected. So I think this is the most important question in our field about the limits of virusphere.
Amazing. Thank you so much, Isabella. It's a fantastic story. I'm sure your family is very proud of you. Kristen, over to you to take care of the business.
Okay. Well, first off, thank you to all the attendees. We have had over 150 questions and comments during this presentation. Isabella will receive them afterwards. So even if we don't get to your question or answer now, or your comment on how much you liked her seminar, which there are many of those, she will see those. So what I've done is I've tried to pick on themes, and many questions have kind of lumped a bunch together so that we can try to get through many of the most commonly asked topics. Before we do, one little business item. Many folks have asked if the slides, recording and notes will be available on the website, and they will be posted afterwards. So you will be able to go back and watch the recording or see the slides afterwards. So Isabella, one major question many folks have asked is, what are the risks of giant viruses to humans and to other animals?
Yeah, I think this is something that people always thinks about, and there is no risks because we don't have evidence of giant viruses being capable of infecting humans or animals. The only thing that we have in literature is some studies that detected some gene or something like this in, I think it was insects and maybe small animals, but we don't have any kind of evidence of the capacity of them to infect and cause diseases in humans or animals.
Another question that a lot of folks have asked, similar vein, is with this superinfection exclusion response that you're seeing, does this affect speciation or coevolution with the host, and does that have an impact on giant virus diversity?
Oh yeah, I wish I could answer this question, but I can right now. I think we have to go a little bit deeper, like I said, about the mechanisms and also about in what this implicates for amoebas and also for the viruses. Like I said, I think maybe we can be analyzing some antiviral mechanism of the amoeba here. Maybe this is not something linked only to the evolution of the viruses, but maybe it can be linked to the host amoeba. So this is something that we have to study a little bit more to understand, but it's always a possibility, but we have to study a little bit more.
Another question that was asked a bunch of times is, do amoeba have native defenses against these viruses? What do they do to keep viruses from attacking them all the time?
Yeah, so the phagocytosis is one of these tries of the amoebas to don't be infected, because after the virus is phagocytized, they are going to be inside phagosomes, vesicles, and those vesicles will be acidified in the cytoplasm of the amoebas. But in this case, giant viruses evolved to escape from this process. So this is one of the ways that they can defend themselves, doing this acidification inside of their phagosomes, inside the cytoplasm, and they have a lot of different mechanisms of defense. And actually, there is a work that I was involved in that showed the mechanism of Vermamoeba vermiformis to defend herself from another giant virus, Faustovirus. So those amoebas, in the presence of Faustovirus, they insist, they become cysts. And the cysts are the resistance form of amoebas. And the viruses are not capable to start a replication cycle inside cysts because the amoeba is inactivated. So there are a lot of different things that they can do to try to defend themselves, not only for giant viruses of amoebas, but also defend from bacteria, fungi, and a lot of different pathogens.
Another question that several folks had is this superinfection defense similar at all to that's seen in bacteriophage? Is there any homology in the genes or any kind of structural homology?
So we still didn't study it, the part of the genes that can be related with superinfection in giant viruses, actually in mimiviruses in this case. So this is also another question that I can answer right now. We have to go deeper also in this part of the work. And this is something that we wish a lot to do in the future, because this really caught our attention. When we are talking about vaccinia virus, we see the importance of those two genes that I cited before. The early expression of those genes are extremely important for this process. So maybe in the future, we can answer this question.
Several folks wanted to know if you could explain a little bit more about the roles of the proteins A35 and A33 and A36. People wanted more clarification on what you were talking about with those proteins.
Yeah, so those proteins are linked to another nucleocitovirus, vaccinia virus, and not with giant viruses of amoebas. And to do the early expression of those genes, it's important for the process of blocking the superinfection of cells that were infected first by vaccinia virus. And they are also linked to that process that I explained before about the actin tail, and this actin tail being responsible for expelling another kind of microorganism outside of the cell after the infection by vaccinia viruses. So they're pretty much linked to this in the process.
You have so many questions. We'll go right up until 2 o'clock. I will try to get as many as possible. I'm loving what I'm seeing here. One question that came up is with the viral factories. Are there specific locations in the cell that these are present in, or are they different locations by different viruses?
So the viral fabrics are always inside the cytoplasm of amoebas. They can be different because of the different viruses that will infect amoeba. So some viral fabrics are more dark in the images. When we are analyzing them by transmission ecton microscopy, and some of them are a little bit more, not so dark, more translucent. And they also can be more organized in some kind of viruses, like is the case of MRSA viruses and mimiviruses, and can be a mess for another type of viruses, like is the case of some viruses like Cedra viruses. So this is the major difference between the viral fabrics, but the locations are basically the same inside the amoeba cytoplasm. And also, I think the most different from all is the viral fabric of Yara virus. It's like something really crazy that happens inside the amoebas after the infection. But I think the location, it's basically the same for them.
Related to that, one of the attendees wanted to know if all of the host proteins are excluded from the viral factory, or if the host proteins contribute to the viral factory.
Yeah. I think, I don't know. I think they are more linked to contribute to the formation of viral factory, if I understood the question. Because after the process of uncoating of the virus, those proteins are liberated inside the cytoplasm of the host amoeba. And then we start to have the formation of an early viral fabric. And this early viral fabric became a mature one. And we start to have all the processes of the formation of newly particles and everything. So I think it contributes.
Okay. One question has to do with, does the superinfection exclusion response interfere with virophages or other co-infecting, bacteria or anything else that the amoeba might phagocytose?
Of course, because when you do the blockage of the superinfection, you are avoiding the competition with all those different pathogens potentially in the environment. So for sure, this is going to cause some intervention for the another pathogen that is trying to get inside. But we also don't can say that we have already studied this before. The only study that we have until now is comparing those three mimiviruses, mimi, moumou, and megavirus. But it's something that we also want to look for in the future. But we wish we can first look for this inside giant viruses only. And we can see already the blocking of the superinfection for bacteriophages also. But I can link those because I never studied this before.
So a lot of people are asking about megavirus not having the same capability to block superinfection. And what are your thoughts on that in terms of the biology? Is that a strategy to promote diversity? Did it simply just not acquire these genes because of horizontal gene transfer? Or is there something fundamental to the biology that megavirus does not have this mechanism?
Yeah, so when we are talking about megaviruses, I studied a lot them during my master's and my PhD. So they are more divergent when we compare the three of them, and we see a lot of differences. For example, like I said, in the surface fibrils, and I studied this a little bit deeper also during my PhD. And I think it can be linked to the biology of those viruses, but I think also it can be linked to the evolution. I think maybe during the evolution, the capacity of blocking superinfection could be something that was acquired for mimi and moumouviruses. And I really think that this is related to the content of the genome of those viruses. And maybe this was not something that megavirus could acquire during this evolution. So I think when we study a little bit deeper those genes, we are going to have so much more answers. And this is something that I am really curious about.
So one more philosophical question before Stacy and I will wrap this up. And that is, with giants being so complicated, what are your thoughts on whether or not viruses are alive or not?
Oh, this is a classic one. Well, I think this is always something that we have to think about a lot to respond. But the canonic idea of viruses being something so simple and linking this to the fact that they are not alive, this is something that I don't agree, because we can see that not only for giant viruses, when we study deeper viruses, we understand that they are really complex actually. So if I think about this complexity, and also when I think about giant viruses and how they are more independent from the host, because they, for example, carry genes linked to translation of the genomes, for example, I think about them more like an organism, like something that is live than something that doesn't have life, because I think about this capacity of, being more, how can I say, being more diverse and being more I think about the evolution of them, and I don't think they are simple at all. So if we are talking about linking the simplicity of them with the fact that they are dead, this is not something that I think it is possible. I think more as something that is alive and complex.
Well, thank you so much, Isabella. This was really a fascinating talk and wonderful questions and answers. For those folks who we didn't get to your questions, I will make sure Isabella can see them all, and we can, if she is able, she can respond to you. And as a final reminder, the video recording and slides will be up on the JVI website after this. Back to you, Stacy.
Yeah, thank you, Kristen and Isabella. That was absolutely fantastic. And just a reminder, when you're going to submit your work, if you're a virologist, think about Journal of Virology. We're looking for everything, and we have a lot of different formats. So definitely lots of opportunities. More information about Discover JVI, at least for 2025, JVI is now open access, which is really important. What's the next slide, Christine? There we go. All right, so our next seminar series is in September, where we're going to talk about herpes viruses. So check out our website for details. And again, this presentation, as well as the past ones, are available on the JVI website. So thank all of you for attending. You can reach out to us if you have comments or questions through JVI at ASM, our social media sites. And we just want to thank Isabella, Kristen and all of you, and we hope we see you in September.